Alright, thank you for joining again, guys. Welcome back to Journal Club. I have something to present today. Does anyone else have things they want to present?
I'll find it real quick. I'll just
Okay,
know when I find it.
okay. Vion, do you want to present your paper, or do you want to hold off on that because it's not published yet?
I can do it in like a week or two.
Okay.
On my computer, the Discord, like I can't talk on my computer Discord, so it'd be difficult to share my screen at the
Oh,
moment.
I see. Okay.
Yeah. But
And
yeah,
Krish, do you have anything that you want to share?
Nothing that I have ready right now,
Okay, okay.
but
That's fine. I guess I will go first then, because Sujan, you are looking for something right now.
Yep.
Okay, cool. So let me share my screen here. Let me share the screen. Alright, can you guys see this Google slide?
yes.
Alright, cool. So I finally have completed sort of my spleen research. I did it for the first study analysis. I'm going to be doing this for multiple studies, and I'm going to be doing it for astronauts' genomes as well. But I am starting with the 506 study from RR23, Rodent Research 23. It was up on the space station recently. We got them back down, sequenced the genomes. And I got to sequence genomes, and I went through Ruth. She's actually in this journal club too. She made programs. I edited them a tiny bit, but she did like 90% of the work. And we made a lot of graphs to analyze the data. And I made gene lists, and I analyzed it, drew conclusions, and can make a statement on what I think the spaceflight's effect on the spleen is. This is one study with an N of 10. No, 27. So it's not like completely conclusive, but I think this is a good start. About the study, no, it's 20. Where did I get 37? Oh, I was thinking of the days, but it's 38 days. It's an N of 20. They were 16 to 17 week old mice. These are mice that have just entered adulthood, so young adults, I guess. And they were maintained in microgravity for 38 days. And there was a ground control study, just in terrestrial conditions. Nothing extra was done to the ground control. So basically, we had these mice we had up on the space station for a bit over a month. And we got them back, got the spleens, sequenced them, and I'm looking at what genes are substantially up and down regulated. Now, what I mean by that, because I know not everyone here has like studied biology intensely. So
yeah.
the genes you have, it's part of your DNA, your genetic code. They express certain things in your body. They tell your body to do or produce certain things that are needed for functions. Sometimes a gene can be turned off or on. And more in more of a subtle way, a gene can be upregulated or downregulated. It can be biased to produce more of that thing or to produce less of that thing. And that can be a problem because the body's existence is a consequence of like a very dynamic homeostasis. It's maintained at a perfect balance, and interrupting that balance is where you get issues in the mechanisms of the body. And when genes get substantially up or down regulated and start producing a lot more than they should or not producing enough of what's needed, you run into problems. So I think there are about 30,000 genes that we sequenced for this. It's just the genome of the mice. It was done with bulk spleen tissue. There's kind of two main ways that DNA is sequenced. One, or that the genomes are sequenced. One is through RNA collection. Well, it's usually done through RNA quantification. And in this case, it's done with a bulk tissue. So we just get like a slice of tissue and we degrade the cells in it and then quantify the genome. And then we normalize the counts because it might be biased if there were multiple different kinds of cells and stuff. And then you can also do it for a singular cell called single cell analysis. I'm going to be doing single cell later, but this is bulk spleen tissue. So we basically just got the genome from a piece of spleen tissue, normalized the counts, averaged everything out, basically. And this is a graph of all of the genes. And this is a logarithmic scale here. So basically the midpoint, you can see it's kind of grayed out there. These dashed lines are 1, so anything beyond these dashed lines is statistically significant. Red is upregulated, blue is downregulated. This is a log scale. I don't know what this gene is. I have a suspicion that this is a error in the sequencing and not actually a 32 a log twofold change of 32, because that'd be insane. That'd be, let's see, 2 to the 32.
I was about to ask. I was like, what is that
I don't
gene?
think this
That's
gene was
kind of crazy.
upregulated 4.3 billion times, so I have a suspicion this was a recording error. But there were some genes that were pretty drastically changed, changed by somewhere in the realm of hundreds. We get some of these here in the like 7 to 8 range. Those are some significant changes, almost complete silencing or extreme upregulation of these genes. I talk about some of them, mainly these two down here, which are a immunomodulatory gene. A gene that modulates inflammation, to be more specific. Not that is the inflammation pathways of the immune system. But basically, the point of this volcano plot is we can kind of get an idea of like to what extent were things thrown out of whack in spaceflight. And we can see pretty drastically, because there is a large amount of genes that all went up or down regulated to a statistically significant amount. What is considered statistically significant here is a change of 2. So either it was doubled in expression or it was cut in half in expression. And then more than that is obviously still kept in. So the baseline is the 1 value, which would be 2 to the 1 because it's log 2. So we also did some pathway analyses. These are not as important yet because I was interested on some specific genes about the spleen's ability to support the immune system. But I'm going to look into this a little bit more later. Not all of this is useful. What this is it's these are signaling pathways that are predefined in previous publications, and there are certain genes that are known to be associated with certain things, but it's not always like actually indicative of what's going on. Do I have it on here? No, I don't. Not all of these are actually relevant to the spleen, and not all of these and not all of the things that it says these genes are related to are what the genes actually do in the spleen. Like on one of my graphs, it said that the gene associated with alcoholism was substantially upregulated. I suspect spaceflight conditions does not induce alcoholism. So there's basically, this is comparing genes to what they're kind of clinically known to be associated with. But that's not a great way to quantify the condition's effect on a specific organ system, because this is ignoring spleen specific. This is just in general what is clinically considered, so not as relevant. What's pretty important here is the analysis of the genes that I was proposing. And there were 2 gene lists I proposed. The first one was this one. And this was centered around immunosuppression, and it was immunosuppression through 3 main things: B cell maturation, natural killer cells, and neutrophils. So basically, these are immune cells that are responsible for doing certain things. Neutrophils, all these cells kind of have multiple roles, but neutrophils are involved in the inflammation response. B cells are the memory of the immune system, and they produce antibodies. Natural killer cells are like a fighting force, I guess you could say. And the spleen, what it normally does, because I don't think most people know what the spleen does. I'll be completely honest with you. I've been studying biomedical. I've been doing BME research for NASA for like 3 years at the point when they came to me for this, and they were like, "Can you do the spleen?" And I was like, "Yeah, sure, I'll do that." And then I went home and I was like, wait, what does the spleen do? So I don't blame you if you don't know what the spleen does, because it's like considered the most useless organ, because you can live without it on terrestrial conditions. But the spleen...
Is it just it's left over then? Appendix or appendix?
I guess you could consider the appendix its own organ system, but there's so little that goes on in there that like I don't even think the appendix comes up in the conversation of organs. Like, I think you'd sooner be talking about bones. Bones actually have a lot they do. But I guess there's an argument to be made for the appendix. Yeah. But The spleen, what it typically does is two things. It supports your immune system. It's not the main immune system thing. It helps support your immune system. If the spleen is taken away, the roles that the spleen did are taken up by the liver and thymus, is what we've seen typically. And it also helps filter your blood a little bit. It's not the main thing for that, but it helps filter your blood a bit. But in spaceflight, things are different. And that's one of the big parts of my hypotheses. I'll get to that in a moment. But to look at kind of some of the effects of the immune system on the spleen, so chemotaxi, I'll define this real quick. In the body, to get cells to migrate or mobilize and move to a certain area, certain signaling molecules are used that attract the cell, kind of like a magnet. It'd be like if the cell was like a piece of iron and your magnet was like a chemotactic signaling molecule. You put it down and it attracts the cell to it. Tells the cell, come over here. So surprisingly, ISG15 was upregulated. I was expecting to see this downregulated. It's a chemotactic factor for neutrophils. Neutrophils, so one of the main things the spleen does is help mature B cells. They're a very important immune cell. They're what creates antibodies. And I think you all know the importance of antibodies in just your general defense against pathogens. It's the memory of your immune system. So B cells, the spleen is kind of like their stomping ground. They migrate from the bone marrow to the progenitor B cells they are differentiated in the bone marrow. They mature into precursor B cells. They mobilize to the spleen. And then in the spleen, they mature through transitional phase 1 and 2, T1 and T2. And then they become matured B cells and they leave. So the spleen, they are born in the bone marrow. They're sent to the spleen. They're raised in the spleen. You can think about it like that. And then from there, they're sent out into the bloodstream to be proliferated throughout the body to do what they need to do. One of the main signaling molecules needed for the survival of these B cells is B cell activating factor, BAF. I don't know why it's not it is BAFF. I don't know why it's not just BAF. But this molecule is essential for the survival of B cells. One of the main cells that releases this are neutrophils. The three main groups are neutrophils, dendritic cells, like the neuronal cells, and monocytes. But one of the main three, the neutrophils, the chemotactic factor for it is upregulated. That's surprising. I wasn't expecting to see that. Everything else is in line with kind of everything else was kind of in line with what my initial hypothesis was, though. We had more upregulation for trying to... We basically had upregulation for trying to support B cells, is what we're seeing here. This is not significant upregulation. These are in the realm of like 2 to 3 times. I actually have ISG1. Yeah, 2 to 3 times. But we basically had the spleen trying to work on overdrive to support B cells in spaceflight conditions. And then in the downregulation, we had, unsurprisingly, things associated with the immune system, kind of suppression of the functionality of the immune system. The spleen is attempting to support the immune system, but the immune system itself is kind of suppressed in the spleen's environment. The actual neutrophils themselves are less abundant, and this receptor associated with neutrophil adhesion. Basically, things associated with the actual neutrophils themselves are downregulated. The spleen is trying to call in more and more of these things, but they're not actually there. This is not enough genes to draw that conclusion. We're only looking at like 6 genes here. So I made a new gene list centered around 2 main things: immunosuppression and circulatory modulation, because it's known under stressful conditions for the body that the spleen can actually take up the role of trying to support the circulatory system. And this is the new gene list, which turned out much better and is much more significant, with some of these genes even being changed by over 100 times. So 2 main things are happening here. We basically, all of these here, except for these, all these here have to do with the circulatory system. And all of these here pretty much have to do with the immune system. GFAP is a glial factor. We'll talk about that in a moment. That one doesn't have to do with the circulatory system. But what's very interesting about this is I came to start to realize it looks like the spleen might be more important for sustenance of homeostasis under spaceflight conditions than previously considered. Because it seems like the spleen is largely taking up the role of other organ systems that are being hurt by the spaceflight conditions to try to sustain our blood and our nutrient transport. So I don't want to go through. I'll go through it quickly. So basically, we have all these genes. So this first one it was upregulated by 20 times. That is extremely significant. And it is a factor for the production of red blood cells. Not platelets, red blood cells. So seeing this upregulated 20 times means the spleen is trying really hard to help produce red blood cells. That's a way you can look at it. Might not be correct exactly. You never know with this kind of analysis, because I just know this gene was upregulated. This merits histological analysis, which I will probably do in the future. But basically, a lot more red blood cells needing to be produced. And actually, before I go into this, does anyone have any questions so far? Because I know I'm going kind of fast.
None so.
Well then,
No questions? Okay.
No, this is looking really cool. Yeah.
Okay, cool. So this gene, I'm going to stutter if I try to say it. I'm probably going to stutter if I try to say all of these gene names. This is the easiest gene name to say here. They're all like just random fucking, just letters and numbers. So this gene is directly associated to erythropoiesis. Erythrocytes is the fancy name for red blood cell. So I don't know why it's not erythrogenesis. Everything in the circulatory system has poiesis at the end for genesis. But basically, this gene upregulated 20 times is my hypothesis. I put it here in red. I suspect it's a compensatory mechanism to try to counteract the spaceflight-induced anemia. Spaceflight does induce anemia, especially because of the stress on the bone marrow. We can talk about that another time, but all the astronauts, it's been recorded in all the astronauts, and we've seen it consistently in many studies. This gene right here, this one is responsible for the lineage commitment of blood cells, not specifically red blood cells, but red blood cells, white blood cells, platelets, just blood cells. Hematopoiesis, not specifically erythrocytes, but hemocytes, blood cells. Hemocyte is a very broad category. So basically, this expression tells us that the spleen is trying to produce more stuff to terminally differentiate progenitors into blood cells. Trying to get more blood cells going on. And again, here we have a upregulation in a gene that is responsible. I think this one is for the structural conditions of blood cells, the structural integrity. Let me confirm that. Yes, this is this one. The knockout of this gene has been associated with. It is that gene, right? Yeah. The knockout of this gene has been associated with some. Well, the complete knockout of this gene just led to death. The mice didn't survive past a weaning age, but the partial knockout of this gene, I think heterozygous mutation, that basically led to the mice having deformed blood cells. They were spherical. Not like that typical blood cell shape. It's like a red blood cell shape. They weren't like this. They were like spheres, which was it hurt their lifespan. Not good. This gene is basically associated with the structural integrity of the red blood cells, it's called spherocytic. Yeah, I did put it here. Okay. Basically, this gene is responsible for producing proteins that maintain the structural integrity of these cells. So, an upregulation in this further supports attempt to maintain a faster production of these cells, or just make sturdier cells. Could be either of the two. But again, we have upregulation of the spleen's attempt to compensate for the spaceflight-induced anemia. This one I think was just the immune system, actually. I think this was, let's see. Oh, I remember this. Yeah, so it's not completely known what this gene does. Some of the genes that we sequence are just completely unknown. Their names are just N/A, because we've not discovered what those genes are. But this gene, it's the leading theories are that it's for moderating T cell response. Basically a immunosupportive gene, and this was found to be pretty substantially upregulated by 11 times, is what I saw in this
How do you
study.
confirm like how do you confirm what these responsible for like in a testing environment?
What this gene is
what the
responsible for?
process of, like, yeah, what is the process for like, yes, this gene is what helps create structure for red blood cells. This gene
So
helps do
you
this.
can
How do they-
artificially change the expression of a gene. You can completely remove it. You can upregulate it or
How do you see
downregulate it.
what happens
Yep.
if you like remove the gene
Yep.
entirely?
Yep. Now, I
I'm
didn't do
guessing they
that.
removed this. They saw no changes, something.
Huh?
So I'm guessing they removed this gene and they saw no changes, so they're still confused.
I'm not 100% certain. What I
And what makes it hard for researchers to not like understand?
Well, part of it could
how
just be
they-
that no one's gotten to this gene yet. It might not be particularly difficult, but it might be that all of our knowledge from this gene comes from like transitively related studies, or that we know things about other genes in this gene family. So like this here, this is a whole gene family, and this is gene 10, and maybe we know things about the 2, 3, and 4 gene of this family, and we're guessing 10 maybe does stuff because we saw in a study where 10 was changed. Along with those, something changed slightly different than the one where 10 wasn't changed. Like, it kind of depends. It might not be that this gene is particularly difficult. It's that there are so many genes, and not all of them have been figured out like what exactly they do yet.
People aren't really focusing on this one yet. Then.
Huh?
People aren't really focusing on some of these.
Yeah. And this one, I don't know if it's like particularly difficult to isolate what it does, because maybe it does something really like small, or maybe they haven't focused on it. I wasn't too concerned about that because I had like 30 genes to get through. So I just found citations that talked about what we know about these genes. So all of these citations are basically just saying what we know. And the NIH is a pretty good database for this kind of thing. This is in the mouse because we studied mice here. But I can tell you kind of what we know about the gene. And I think this is sequence notation. Yeah. And where we found the gene and studies that they got this information from. And so this is a good place to get gene information. And then I try and find actual publications that talk about it. But yeah, this is the only gene I have on here that's not like completely like it's iffy, but it seems to be related to moderating T cell response and activation. So it's an immunomodulatory gene, and again, it's upregulating this to try to get more immune system activity going on. But I don't think there's as much. I don't think there's as many immune cells. I really should try and get some blood samples from the astronauts to take a look at like the counts of these things. There's probably data on it. But yeah, this one not as interesting. I can't draw as big of a summary on this because it's not directly known. This gene, again, we're back to blood. Lots of stuff about blood. This one is for ion transporters for blood cells. So basically on the membrane of red blood cells, this is an anion transporter. It's an anion exchanger. So basically it sends nutrients in and out of the cell. This is similar to the upregulation of that structural integrity gene in the sense that like it could maybe be that the red blood cells are having more anion exchanges, but I suspect this is to try and keep up with the attempt at producing more in that localized area. Like if you're making more cars on an assembly line, you need more wheels kind of thing. Not that we're putting more wheels on one car. But again, more and more of these genes related to the maintenance of the circulatory system are being upregulated in the spleen, which was something that I had not seen anyone talk about in any of the papers for the spleen in spaceflight, because there was only one paper for the spleen in spaceflight when I started this. So that's why they had me do this, because no one really knew the effect of spaceflight on the spleen. This one is interesting. This one's very interesting because one of the main things I was worried about was a decrease in the ability to support the B cells in the spleen. And one of the things we'll see down here is a lot of the chemotactic molecules for the neutrophils are actually substantially downregulated, except for ISG15. For some reason, ISG15 is slightly upregulated, which we talked about up here. It's like twice its expression. But all the other ones are like suppressed by like 10 to 12 times. So, the neutrophils, one of the main sources of expressing the B cell activating factor, they are the chemotactic molecules, the things to tell them to come here. They're all pretty much substantially downregulated, again, except for ISG15, which is slightly upregulated. So I'm guessing that there's far less neutrophils in the spleen, because also the genes related directly to neutrophils are decreased, suggesting there's less neutrophils. So that B cell activating factor, the BAF thing that we talked about, that's necessary for the survival of these cells, well, less of it is being produced, and we need the B cells. So I think this is a compensatory mechanism from the peripheries of the nervous system to try to innervate the spleen with something that can take up this lost responsibility from the decrease in the neutrophils. Basically, the main glial cell for the excretion of B-cell activating factor are astrocytes. A pretty cool sounding cell. I'll show you a picture of them because they look kind of cool. This is why they're called astrocytes. So there was a big 11 times upregulation in the main protein responsible for astrocytic activation in the spleen. I suspect, now this is not enough evidence to confidently draw this conclusion, but I have a hypothesis that this might be increased innervation of the spleen through the astrocytes, being the main glial cell that produces B-cell activating factor, to compensate for the loss of BAF expression from the other main sources, being the neutrophils and the monocytes, which are downregulated. And even a lot of the chemotactic molecules are downregulated. There were two up here that were slightly upregulated, but there's some down here that are substantially downregulated. So I think this is an attempt at the body to try to get more of that B-cell activating factor into the spleen, to account for the loss in the other cells. But I can't say for certain yet. This merits histological analysis as well. So we'll find out eventually.
And these changes are pretty, like,
They're big.
I mean,
They're big.
from perspective, that's not a biologist. Like, these seem like pretty massive changes.
These are massive changes. Yeah, no.
Your body is pretty like sensitive to changes in temperature, pH, like even like connectivity, right? A lot of it based like models and like cell transport and whatnot. From what I understand, it's based on very, very acute changes in things. So these like upregulations, downregulations being the scale of like 10, 20, how does that not throw all your whole body off balance? Like, it's not. Or does it?
So it does throw major systems off balance. Bones start to degenerate. The astronauts and the mice and everything on the space station starts to become.
So this is related to the osteoclasts being produced and the osteoblasts being like not produced anymore.
This is related in the sense that the bone marrow is where the progenitors of the immune system lie, and it's that very bone marrow that gets screwed by the microgravity because it's so mechanosensitive. So a lot of the immunogenesis, that is the foundation of the sustenance of the immune system, is hurt a lot because of that, because of how hard the bone marrow is hit. And like these, what we see is many failure points of the body in spaceflight. Spaceflight is not safe. You get the degeneration of your bones, you get anemia, your immune system becomes extremely suppressed. There's a lot of stuff that goes pretty poorly on the space station.
You're saying the spleen is essentially trying to compensate.
That is what I'm discovering here.
Yeah. So, but I'm guessing other organs also have some level of compensation to them as well, right?
Yeah, that would be my guess as well.
Like, for example, if you're in true microgravity, I'm assuming your heart doesn't have to work as hard to pump blood through your whole body, right? So does that affect like your blood pressure? I mean, I'm sure it does. Like because if you have blood
Yeah,
pressure
it would have
super
to.
low, that also like messes with like nutrient transport. No,
Yes, it
um,
absolutely does.
interesting. So
I
which of these the gene that's responsible for creating osteoblasts? I'm guessing that's downregulated then in spaceflight, right?
Okay, so that is a
Osteo-
good question. That's kind of the main work we're doing at the bone signaling lab. One of the main things we found is that it's not a gene responsible create. It's not a gene responsible for creating this downregulated, though that might be happening too. But one of the main things is a gene typically turned off that produces a inhibitor molecule is drastically ramped
Oh,
up.
okay.
and
That
and
inhibitor
and it's
prevents
it's it's it's
osteoblasts.
mechanical loading that typically that typically suppresses that inhibiting molecule.
Interesting. It's almost like so then like in a sense that seems like the body has it kind of has a way to regulate like massive changes in physiology, like if weight to change heavily or
I mean,
because
yeah,
that's the only reason like
the body's ability to maintain its function is extraordinary. Like you can
yeah,
have
but I'm just the reason it might have the whole mechanical load, like I guess the reason we might have that is to compensate for massive changes. Like
yeah,
I don't
yeah.
know, if you were like an ancient human, you lost like an arm, maybe the body would like slightly adjust your bone density so
I
that you move
suspect
on.
it might have to do. Maybe it goes as far back as the creatures that evolved out of the ocean, and the change and the mechanical loading on your body under those situations, depending on the
All going from
yeah.
I guess that makes sense. From water to land.
I mean, who
Hmm.
who who knows exactly, but yeah, there's a lot of it's amazing how the body's ability to account for extreme changes is extraordinary. Just the very fact we have regenerative ability is like insane, and that we can and that we can sustain ourselves by consuming all these different forms of nutrients, and the body can consistently break it down and reallocate it to it. I mean, it's the like down to the molecular interactions. It's unfathomable, like the level of complexity that is at play in just a few seconds. All we can hope to do is like isolate individual systems and look at their interactions to their local environment. But I mean, with the computing systems that are starting to be produced, we might get to a point in our lifetimes where we are able to simulate true physiology. That would be insane. But yeah, this one I suspect is another compensatory mechanism, and to account for the decrease in the BAF expression. Oh, we lost someone. We lost Vion. Here, let me message her. Well, for now I will continue. Let me do that. I'll see if she responds. But yeah, so I suspect this is a compensatory mechanism for trying to account for the loss of the expression of B cell activating factor. I can't claim that is what this is confidently, because that's a little bit of a stretch for the evidence here. I need to look more into that, but I think it's possible. And then for downregulation, the least downregulated molecule here on this list is IRG1, downregulated by 6 times, which is still very significant. And this is something for the immune system. This was, let's see. Oh, okay. It was the macrophages. So this is a anti-inflammatory agent. Basically, something to quell inflammation in localized areas. The expression of this was downregulated 6 times. one of the things we have seen in spaceflight as a consequence of the suppressed immune system, we already knew about immunosuppression, just in the context. Oh, welcome back, Vion. I assume that's Vion. Yes.
Yeah,
Venus.
I
Yes.
got booted off for a second. My dad called me.
All good.
Okay.
All good. So we just moved off of the this one that we were talking about. There was a compensatory mechanism for the B cell activating factor, and we're now on the downregulated genes. The genes that we saw were expressed way less than they typically were. And the first one is an anti-inflammatory. It's a enzyme to activate an anti-inflammatory response in an immune cell called a macrophage. And basically, one of the things that we know in spaceflight is there's extreme immunosuppression, and to that end, one of the consequences of this is persistent inflammation that the body doesn't have the ability to quell. And that's stressful for tissue environments and can inhibit other forms of the homeostasis. So it's a pretty bad thing. What we didn't know about the spleen was what exactly was suppressed and what was what happened in the spleen specifically. In the spleen's environment, what I found is it's this gene for macrophage anti-inflammatory response. So this gene is downregulated 6 times. So that is definitely going to contribute to the persistent inflammation that we see. Also, glycam-1 was downregulated by like 6.5 times. This was for general immune system sustenance. I believe it is in some sense related to chemotaxi. It's a molecule that can help migrate white blood cells. So basically, this gene is associated with getting white blood cells to move throughout the body where they need to, mainly move out of the blood into the peripheries. And having this downregulated means more of those white blood cells are not going to get to where they need to go and just kind of maybe stay in the blood. This one, downregulated by 8 times. What does this do? I don't remember these genes by name. There were too many that I went over. Oh, yeah, this is responsible for, I think, an antimicrobial. So this gene is basically it's surprising to see downregulation of this in the spleen, because it's not typically recorded in the spleen to begin with. But this one is responsible for kind of an antimicrobial defense, and it is its downregulation means you're just more susceptible to microbes. So pretty bad thing. More immunosuppression. Here's 12 times downregulation, and this is a main chemotactic molecule for neutrophils. So this is the ligand that tells neutrophils you need to come here. And if you remember, they were the one that was responsible for excretion of BAF in the spleen. They were one of the main molecules of dendritic cells, neutrophils, and monocytes. So seeing this downregulated by 12 times suggests that there's a lot less neutrophils being recruited to the spleen. And I found that the clusters of differentiation associated with the neutrophils, basically the ways we can identify were there neutrophils in this area, those are also downregulated by like 4 times. So there were definitely way less neutrophils in the spleen. And this is probably one of the reasons why, because the main chemotactic agent, the main chemoattractant for neutrophils, was downregulated by 12 times, which is a pretty extraordinary downregulation. And the two of these that were astronomically downregulated by 175 times and 123 times are inflammatory or are inflammation modulation proteins. Now the downregulation of these by such a large amount is somewhat proportionally consistent to the natural upregulation.
175 times.
Yeah. It was like a 7.58 or something. But in normal conditions under extreme inflammation, these can actually rise in the blood 1,000-fold. So that their expression is in a much higher concentration, which I think accounts for this increase in proportion of downregulation, seeing as we went from 12 times to 175 and 123. I think this molecule is just expressed in larger quantities typically. What these are a molecule that, so clinically these are the main markers for inflammation that we've been using in like a hospital setting. Their exact role is a little complicated and was a bit nebulous in the papers I could find, especially for mice. There wasn't anyone confidently saying what they did in humans, so then for mice there was a lot less. But basically these can either these can both increase and decrease inflammation under different circumstances. But basically the consistent theme with these that I saw was they are expressed in response to inflammation in the body. So from pathways that identify inflammation, these are upregulated. Now, for some reason, these are extremely downregulated in spaceflight, and we know there's persistent inflammation in spaceflight, so you'd expect to see a lot more of it. But it's actually downregulated compared to the baseline, not even compared to what we would expect. So I suspect the pathways that are capable of identifying the inflammation are silenced, and that also would explain why we have such persistent inflammation that is unaddressed. So I think this is again a consequence of the spaceflight's effect on the pathways for identifying and modulating the state of inflammation in the body. Basically, my overall conclusion, I won't read this word for word, you can read it if you want, but my overall conclusion is that the spleen might be more important in spaceflight than we previously thought. It seems to take up a major compensatory role in sustenance of the circulatory system that we had not realized. And also, unsurprisingly, we have substantial immunosuppression as a consequence of the spaceflight, stressors. And what I have pinpointed those two are two main things: being decrease in the chemotactic molecules for a lot of the immune cells, kind of loss in the ability to help transport them where they need to go, and also, oh, hold on. Sorry, I'm back. The second main thing being a decrease in the ability for the spleen to serve as a place for maturing the immune cells because of loss of a lot of the molecules it needs to get them there and to sustain them. So the two main parts of my hypothesis are taking up a large role in sustaining the circulatory system, and the specific pieces of immunosuppression being decrease in chemotaxis and loss in ability to help mature B cells, natural killer cells, and those sorts of progeny in the spleen. So yeah, that is my analysis on the spleen in spaceflight. Does anyone have any questions?
Nice. So right now, Pia is reviewing your paper all written. All figures done.
She's reviewing this presentation. That's why I put this note here.
Gotcha.
Yeah.
And so you're what is it? When's your deadline for paper?
29th.
Oh shit.
Well, for no. 29th.
Coming up.
29th is for an abstract.
Oh.
Oh wait, when's the actual paper due?
I don't know, because I don't know what conference she wants to try to submit it to. I think that's going to depend on how big she considers this to be. Like, if this is not that big of a deal, she's probably not going to want to try to submit it to like some big journal. But if it's actually pretty interesting in her opinion, maybe she'll want to submit it to like a bigger journal or something. I don't know.
Like, are you doing a conference paper or a journal paper?
So I'm going to be presenting an abstract at a conference is the plan, but I have to get accepted. And then a paper
are you submitting to a journal or a conference? Like
both.
Like are you doing a 6-page paper or are you doing a 30-page like document? Like,
well, you don't need to make a 30-page paper to submit to a journal. That's like a lit review.
By a journal, like a journal paper. A journal paper is like a specifically like long form research paper.
I've never heard of that. In biology, it's usually just like you have your typical publications and you have your lit reviews.
Interesting. Good.
I might just be ignorant though and just not have realized when reading papers. But I've never seen a non-lit review reach like 30 pages.
Yeah, there's some like we have the conferences we all do conference papers. It's only 6 to 8 pages, right? Um, but yeah, interesting. Spleen important. Nice.
Maybe. I want to do this for more studies, but that's what it's looking like.
Do you want to keep pursuing this next year, this year as well? I mean,
My
when's
main
the
interest is in organogenesis mediated through the bioreactors I make. That's my main research. This was just a secondary thing. This kind of just happened because I went to my PI and was like, hey, can I try and like publish my own papers like first author? And she was like, well, we have the spleen still that's kind of unknown because no one cares about it, so you can do that. And I was like, okay, sure. But my main work is with the bioreactors.
Is she back now? Like, is she like
Yeah,
responding?
no. As of like 2 weeks ago, she's finally back. So that's kind of why
Oh,
I've been so busy all of a sudden, because she was gone for like over a year. Now all of a sudden she's back, and then she was like, hey, by the way, deadline 29th. And I was like, oh, what? That's in like 2 weeks. That's crazy. So I had to start working pretty hard on this. But this came together in like a week. I was working on this for like 6 hours a day. Most of it was I made a whole new gene list, and we had to make a program to actually plot all of these. Ruth had the expertise for that, though. Her knowledge on that.
map plot?
Huh?
Is that map plot?
This is just R.
Oh, I forget you guys use R.
Yeah.
Weird. Odd.
Yeah. I
Odd
was
coding. Like
saved.
biologists.
Ruth saved me so much time because before coming to RPI, she had like the sole thing she'd been doing for like I think like 6 years before coming to RPI was doing omics research at a lab at Cornell. Though it was like it was remote because omics research is all on a computer. So she had all this experience with making these softwares. So she was able to put something together for me to like just change to fit what I needed in like 2 days. Had I had to figure this out on my own, it probably would have taken another, like, with ChatGPT, maybe like a week or something. If I wanted to understand every line of code. But yeah, does anyone else have any other questions?
I wanted to ask something. So
Yeah.
you mentioned a little bit later about there is an upregulation in GFAP for I think a little bit down. Yeah,
Yes.
you mentioned that upregulation in GFAP. So you mentioned that it was for neuroimmune interaction increase, but I was also wondering because in a lot of my research we use GFAP as a marker for inflammation.
I saw that.
As
Yeah.
So I was wondering if like because you mentioned there's a lot of anti-inflammatory sort of or there's a downregulation of a lot of anti-inflammatory genes
Mm-hmm.
in the sense of like trying to reduce the or there's a reduction in overall like sensitivity of the immune system to inflammation. Um, so I was wondering, do you think that GFAP is playing a role where it's not interacting with inflammation at all in this instance, or do you think there might be some process of the spleen trying to upregulate GFAP as a response to inflammation because these other pathways are being either downregulated or inhibited by some issue regarding microgravity?
That's a good question. I had forgotten about GFAP as an inflammation marker, and it probably, I suspect you're probably right. It's more likely that this is in response to inflammation than playing a unique role. But I did also find out that the astrocytes are the main glial cell that are responsible for BAFF. So
Mm.
that seemed a little like coincidental to me that all the stuff relating to BAFF was downregulated, and the main glial cell responsible for BAFF was its main activation gene was upregulated 11 times in the spleen. So it's like, well, that's interesting. That's
Yeah,
how
that's very specific, correlation.
that's why I put the caveat. Did I write it in here? I wrote it down here, I think, on this one. Like, that's why I put this caveat that, like, this is a stretch, given this current evidence.
Mm.
But that part might be interesting to look into.
I mean, what I was gonna. Oh, sorry. Go ahead.
What I was just gonna say was like, because we already know that there's a lot of inflammation, and we've seen consistent increase in all the inflammation markers. So if there's an interesting part to this for my research, it would be the compensatory BAF mechanism, which I would assume it's not, but I think it merits some histological analysis of like the innervation and the BAF expression.
Right. Yeah. Yeah. Definitely. I think that is definitely a good point because it's like it's so specific of like if it's only like it's a large majority coming from astrocytes, it's definitely something that's like it could be. I mean, because GFAP isn't the only. It's not only an inflammatory marker. There are other things that it does. So entirely possible. So yeah. I mean, it's
Yeah,
cool.
that's a good question. Are there any
Thank
other
you.
questions? All right. Well, in that case, thank you for listening to my presentation. Sujan, do you have something you want to show? Oh, Sujan is deafened. We have lost Sujan.
I'm so sorry. I actually have to drop off now, but yeah, I'll see you guys next week or so.
Okay. Sounds good. See ya.
Yeah. Bye.
Unfortunate timing. I might also have to go pretty soon.
Okay.
Usually it's only the hour I can do, but I guess it's just I just want to say like your research is really interesting. I didn't realize like I just thought it was like very interesting about like the regulation, how much it is there, and also just how much the changes are, the fold changes on some of these. Like the SA 1 and 2, that's a crazy fold change.
Yeah,
Like
I think that's because that molecule is typically like seen to have a change in response to inflammation that's much larger than its other like genes in its family.
mhm.
Like in
Okay.
the literature during inflammation, like it can rise up to 1,000-fold is what I found.
Okay.
So I
So you
think
think that's just
yeah,
mhm.
I think this is an analog to like this kind of like 12-fold to 15-fold increase. It's just that SAA
Right.
itself is expressed in larger quantities. So I think that
No, yeah.
like because it's being compared to ground control where there shouldn't be inflammation, so there should be close to zero. But then it's then zero sets the baseline, so then it increases by huge numbers compared to these because these are normalized. So
Mhm.
this doesn't account for the fact that SAA is expressed in much higher quantities than most other molecules.
Gotcha. Interesting.
But yeah, I think that
Yeah.
like tenfold changes, those are pretty significant.
Yeah.
I think that's where the big
Hey,
things are.
I was back.
Welcome back, Sujan. So Vion
I
has
had
to
that
go.
would be. I can call on the way down back to La Jolla. I'm going. I was just up here for a bit, but
Do you have to go now?
I can stay and call. I'll just be in the car while I'm calling.
Okay. Well, we
I,
might.
I was. Yeah.
I was just gonna say we might end it here then, because you were
Okay.
the only other person who had something to present.
I could. So I'll do a more proper presentation either next week or the week
Okay.
after. Uh, but essentially, like, I'll put the paper now so you can look at it. It's
Okay.
pretty interesting.
We'll do like a
Um,
preview.
yeah. I'll. There's no place to put this. I'll just put it in my little corner. It's not astronautics, but TRI is like one of the most. It's like a pretty big robotics institute, one of the biggest. These guys bought Boston Robotics, so Boston
wow.
Robotics owned by these guys as well. So they're pretty thing. To work for them, you need a minimum of masters, so it's kind of fucked. But you know, it's still pretty interesting. But like a lot of robotics research right now is basically using learning to do like
I've seen that.
uh, a bunch of like unprompted tasks, like untrained tasks, right? So like let's say the table in front of you, right? There's a bunch of things you can do, like fill the pitcher, organize the balls into different bins, uncap the pen, right? Very like human tasks. So kind of like that's like a multitask, right? And it requires more fine manipulation, like not super fine, but like not like typing on a keyboard quite yet. That's actually quite easy with tasks. It's more like you can sort things, you can put dishes away, you can fill cups, empty them, dry them, stuff like that, right? A big issue is like it takes a lot of data to use, and even then, the actual task completion rate is pretty low. Because a lot of people use what's called single task training. So, like, let's say you have a table full of everything, right? Like dishwashing, cup filling, whatever. When you do this training on a single task, and you actually if you go to page 9 of the PDF, you'll see there's like a really cool like graph. And what these guys figured out in like when you use the same system, instead of having like a ton of data, like you had a ton of data for single task and training everything on that, just training on 15% of the LBM fine tunes. LBM is their own model they use, and they're kind of the idea behind it is instead of training on a single data set, they're using like data from all the tasks, right? So you need to tell the robot just to like, sort the vegetables. Like I've trained it on everything, only 50% of total data versus doing only single task data. And you can actually see that the success rate of the model with training a small section of the full data has a higher success rate and a higher completion rate than using single task data for the same trial.
Do you know how they are, like what type of data is being pulled from? I assume it's from image processing.
Yeah, it's just image processing and
maybe coupled
yeah.
with like sensors or gyroscopes or something, but
it's a huge transformer. It's called a diffusion transformer. It's actually there's a diagram of it in page 12. These are like it's like a massive dimensional array with inputs of vision. Like they use some of these sound, dexterity. It's a huge like. To be honest, I don't think the researchers themselves fully know how that works. Right? Like AI has gone to the point now where like the architecture is so advanced that no one person will actually knows what's going on inside under the hood. Right? It's kind of like you have all these inputs and all these outputs. The actual interplay is the billions, like interconnections. Like no one actually knows truly how it goes. That's why like a lot of AI researchers are just using the model and seeing what it does, right? Like for example, for a while when you ask ChatGPT to do math, it would like translate between Chinese, Greek, and all these other languages because I know people couldn't figure out why, but it turns out like in those languages, it's easier to use certain concepts and understand certain concepts in those languages, right? Like time in Greek is like more volume based, and it's kind of like a tangent, but like it would use other languages like while doing math, right? So things like that's like you don't truly know what's going on in the hood, right? But this is basically that they said, okay, here's architecture for having all these inputs, outputs, and to do some learning of these images, right? And instead of just having a single task and a single training data, they gave all of it and only gave it a small percentage, right? It takes longer to train, a lot longer when you have all data, right? And a lot of it's like in a sense almost wasted. Someone might say, or someone used to think, because if you're just learning to sort cups, why would train on sort vegetables, right? But by doing that, it actually had better performance, right? And like their main theory, it's very jargony, but their main theory is that in their training mixtures, like because the way they create the training mixtures is like a statistical analysis of a bunch of different parameters. But what they think is that like the learning vectors of the robot, when you have a set of like multiple tasks in front of it, it kind of like knows not what to not do, right? So like if it's telling you to sort, if you're training for sorting vegetables and cleaning dishes, right? It knows that it can kind of more easily distinctify between the different tasks, and that's like a solid hypothesis for why these models do better with a more diverse data set, right? And this is actually started being implemented in labs across the country, right? Already, like this is pretty. A lot of places have started using like multi-faceted data sets instead of single data sets, including my lab, um, for like robotics research and handling, right? The biggest thing right now is manipulation and handling. That is huge, right? It's still like pretty far behind, but this is a good step forward. I'll go more in depth into it because it's very, very like heavy jargon, like math
This
wise.
is cool though.
Yeah. And transformers are a bit hard to understand at first too, so I'm still getting my head over those. But yeah, it basically shows that like theoretically it's better to train it with data that you might not even use for like certain tasks, right? Um, yeah, and it does basically like using LBMs and using all data sets takes a lot more time, right? It scales like exponentially. That's why they use only a percentage of data set, right? But yeah, it's quite interesting how they set it up. It's not something you yeah.
Sorry. Did they
No,
make this like open source and available?
No.
Okay, I didn't think so. So,
But I mean, this is not like this model. I mean, they have the architecture already in, so someone who has knowledge to do this could replicate it. It's like they didn't do anything novel like hardware or software wise, like that. It's just like a different application.
I understand.
This is more of an analysis of like a model change they did and like explaining why it makes sense.
I imagine one of the important things is putting like the weighting on what percentages of data sets it'll pull in pursuit of like what training. Did they address that at all in here? For like an optimal way to approach that, or?
I'll read more on the paper.
Okay.
I didn't read the yet, so yeah. That's why I'll get back into that.
Yeah. This was just
it's a very influential paper. So,
this is cool. This is a good find.
Yeah. Did I tell you, James? I'm actually not applying. I'm probably not applying to most PhDs this year.
Yeah, I remember you telling me. I'm so upset that this is happening. When we graduate high school, it's all screwed. When we graduate college, it's all screwed. Like that's
yeah,
so frustrating.
I think go for a job/masters for now. We'll do a job for 2 years and then masters. And then I'll see how it is from there.
I'm just hoping RPI will accept me.
I mean, you have RPI, right? For
Hopefully.
At least safety. I think you'll be fine. I think you've given them enough work, in terms of data.
In New York, James?
Huh?
Are you sure? 6 years in New York weather?
No, but I want to pursue a PhD.
But why not also just do a job for a bit and then do it afterwards?
Because I'm not going to get the kind of experience I want from a job.
There's a lot of good R&D jobs, especially in biotech in like Boston, for example. You should ask Saurav. He's not biotech or bioengineering, but like,
I don't know. Just my experience working for Medline, at least, like it wasn't like.
Medline was different because that was more of a production line, like.
Yeah, I think that's probably what it is.
Like that wasn't or does the same sense like an R&D company per se, right? You have a lot of papers out. I think. What's your GPA? Like 3.8, 3.9?
It's like 3.8.
Yeah, so like you can also apply to your program. Like, I'm sure if you apply to the UCs, you get into one of the big 3. So,
Yeah, well.
I just think
we'll see how it goes.
I think I'm going to submit my 2 papers for now, and then I'm going to leave my lab for a bit, and just explore, like, do my own stuff for a bit, and then see how that goes from there. 'cause I like research, but to be honest, James, like at least from the engineering side of things, like paper writing is so like
I'm with you. Dude,
so
I
so
lose my
monotonous.
time every time I write an abstract and have to talk about like
So,
like they just want like the most general stuff to be restated every time. It feels like such a
yeah.
waste, especially the
Well,
bio papers. Like they'll drone on for like pages and pages and pages about.
the thing is, like, what sucks is that the people who are actually going to use these papers are not reading all that. Like,
They're
all
not.
people read is abstract, the figures, conclusion, and then maybe they'll read the rest of the paper,
Dude,
right?
we
Mm.
we need to start like taking a page out of the physics journals. They have it right. They just have like 3 pages. Just like here's the model, here's our finding, here's our conclusion. 3 pages.
Like, I realized like doing 5 years of this like would drive me crazy.
Yeah,
I did
the
2 months of actual paper writing, and I went crazy. So I do want R&D jobs. I do want a master's at some point, right? But I don't think I can do it like commit to 5 6 years of a PhD.
my thinking is I'm
Also,
willing to put up with the bureaucracy if it means I can work in a lab and kind of pursue my own research.
Well, I feel like a lot of that just comes down to you have to find a good lab when you are looking.
Well, I don't think whatever lab you pick, you're still gonna have to deal with writing
Well,
papers
yeah,
how your
but
PI wants you to, and how the journals require them, and
the better the lab or the better the PI, the more easy it is to like communicate because there are some PIs that are just like won't tell you anything and you go to them and they're like, this is not how I want it at all. And you're like, okay, well,
yeah, that would be a another level on top of what I've experienced. I haven't had that yet, luckily. All right, well, I think I got all my questions answered for now on this transformer thing. It wasn't the full presentation yet, so. Is there anything
I'll
else
take some
anyone wants to
notes.
say before we finish this journal club meeting?
good.
All right, cool. Well, thank you again, guys, for joining for Journal Club. It was nice having you. So at some point here, pretty soon, I'll do a reschedule, maybe in like a month or so, once everyone knows their upcoming schedules, because things are gonna change again. But yeah,
I think probably weekend's not the best bet for journal club. So I
yeah,
think like
we'll just see what works best for everyone. I'll put out a
especially
poll.
the fourth year, I'm gonna be out. I'm not out, but like busy most weekends. Like but even summer journal club is hard to make it to because most Saturdays I'm like doing stuff. So
All good.
ideally, I mean obviously we set schedules up, but like I do like a school night. That's like the least busy school night would be good.
Yeah.
I'm trying to get back into actually getting into more journals again and reading papers and doing that. I kind of slacked this summer.
I've been doing good on physics, but I've not been reading any BME stuff really.
Much robotic stuff. I was learning about the interactions of quantum chromodynamics and
Yeah, yeah. No, I was
the
sitting there reading about like new
red infrared
metamaterials.
flares. Yeah.
Like, oh yeah, I'm definitely gonna use this exciton polariton metamaterial in my organ on a chip. That'll be useful. I
You never
didn't
know.
get myself to. I've been losing my mind reading bio papers because they're like 15 pages of stuff I don't need to know. And physics papers are so nice. Like, I can learn so much in such a short period of time. I don't know why it has to be like a textbook every time I want to read about some new finding in biomedical engineering. Oh well, what can you do? Alright, well, I'm probably gonna make like some unofficial like YouTube channel or something, and then I'll just post these videos on there, and then people who miss the meetings, they can see it if they want. But yeah, it was nice having you guys. Have a good night.
So
As well. Good night.